Jessica, Alexandra, Emily, and Isabel. Today we are going to analyse the study, Bovine Embryo Development After IVF with Spermatozoa Having Abnormal Morphology, conducted by researchers within the Department of Dairy Science and Department of Clinical Science of Virginia Polytechnic Institute and State University in 2004.
This investigation utilised semen samples collected from 6 Holstein Friesian bulls before and after scrotal insulation. The principal aim of this study was to evaluate effects of scrotal insulation on morphological features of bovine semen and how this affects fertilisation and embryonic development following in vitro fertilisation. An elevation in testicular temperature by thermal insulation or increased ambient temperature of the scrotum consequently leads to disruption in spermatogenesis. This study was undertaken to analyse and determine any correlation between abnormal sperm morphology and embryonic development. It was hypothesised that an increase in the scrotal temperature of the bulls will affect sperm morphology, impacting on fertilisation and embryo development. The null hypothesis generated was that there would be negligible effect of scrotal insult on sperm morphology, and hence fertilisation and embryonic development would be unaffected. The investigation takes into consideration that there are variabilities in the effect of different sperm morphology on fertilisation and embryo development. These factors are largely dependent on individual characteristics of bulls, including sperm capacitation ability, overall fertilisation rate, and embryogenesis. Semen was collected multiple times from each bull, allowing for within-bull comparisons to be made on 4 of the 6 bull semen. This methodology allowed for more accurate and extensive analysis, rather than simply studying semen from one bull. In comparison to other studies, results from this investigation presented new findings in relation to the variable levels and types of abnormal sperm morphology and their effects on fertilisation. Previous studies have described how piriform heads are associated with subfertility in cattle, and that abnormal morphology can be related to impairment of sperm motility. Another study suggested that abnormal spermatozoa are able to bind and penetrate the zona pellucida, leading to decreased fertilisation rates due to developmental defects. This study determined a significant negative correlation between the number of piriform heads, diadems, asymmetrical decapacitated sperm, and apical vacuoles and embryo development. Hence, this study's findings reinforces that abnormal head shape has a detrimental effect on fertilisation and embryogenesis, providing new information to utilise in sperm examinations and semen quality testing in the future. This is an important area of research as conception rates are a major limiting factor affecting both beef and cattle industries today. In assessing any conflict of interest, it should be taken into consideration that the study was conducted on Holstein Friesian cattle, predominantly used in dairy production. When extrapolated to apply to other dairy breeds or beef cattle, such as Brahmans, findings may not be accurate. The authors may also be biased coming from a dairy background university, with their primary focus being on assessing fertility of the Holstein Friesians.
Experimental group with 6 Holstein bulls, but only the morphology of 4 bulls was analysed, and 400 cow oocytes obtained from a slaughterhouse. The intervention in this study is scrotal insulation. The way the scrotum was insulated, however, wasn't explained in the study. This is measured by collecting ejaculates twice weekly, starting at 8 days to 34 days after scrotal insulation. These samples were used to analyse the sperm morphology such as piriform, tapered, asymmetrical, and severely misshapen head shapes, and nuclear vacuolization. The ejaculate collected between days 13 and 34 after scrotal insulation will represent spermatozoa that were undergoing spermatogenesis during scrotal insulation. Thus, only the ejaculate samples collected 13 and 20 days after scrotal insulation were used in IVF. 8 days after IVF was done, each zygote stage of development was noted, as by 8 days the embryo should be a blastocyst. An ejaculate sample from 5 days before scrotal insulation is also used as a control. This sample size is reasonable for comparing pre- and post-insulation sperm morphology within individual bulls, but wasn't big enough to say that sperm insulation caused abnormal sperm morphology in all bovine bulls. Thus, there could be misinterpretation of data. The sample size was big enough to compare embryonic development in an individual bull's samples. The several flaws of study design include not mentioning the method they used to insulate the scrotum, why they only included 4 of the 6 bulls for IVF, not providing enough information on the cows, oocytes, or bulls, not monitoring the bull's behaviour during the study, and not considering that sperm with normal morphology could also cause abnormal embryo development. We're left with no information on how much human error is likely in this method, and thus left with no idea on how much of the results are possibly affected by this. There was also no explanation on why 2 of the 6 original bulls were excluded. This should be questioned because the results excluded could have massively impacted the results of the study. There was also no information given on the oocytes such as age, health, nutrition status, breed, and environment the cows were kept in before the oocytes were collected. Thus, a conclusion cannot be made that abnormal sperm morphology alone causes abnormal embryo development. Information on the age, health, nutrition status, and environment the bulls were kept in during the study also wasn't provided. This would also affect sperm fertility. For example, if the bulls are kept outside in a paddock, the temperature and behaviour isn't controlled. Environmental temperature could cause an unintentional insolation, and inadequate diet or trauma could affect fertility. The last flaw identified in the design was that the authors didn't consider that sperm with normal morphology could also cause abnormal embryo development due to containing damaged DNA. This study has several factors that cause bias towards supporting the hypothesis. These have already been mentioned but include sample size, oocyte fertility status, not considering human error, not including all bulls in IVF, lack of information on the bulls, and the possibility of sperm with normal morphology to cause abnormal embryos.
The main outcomes of the study were that scrotal instillation is indeed an effective method of obtaining semen samples with high percentages of abnormal spermatozoa. For bull 1 and 3, normal spermatozoan morphology greatly decreased from pre-insult to 3 weeks post-insult. The paper does not state the decrease of normal spermatozoa for bull 2. It can be seen that bull 1 and 3 were the most affected when compared to bulls 2 and 4, and therefore larger embryonic responses are seen from these 2 bulls. For bull 1, embryonic development significantly decreased post-insult, whereas bull 2's embryonic score increased post-insult. Bull 3's development score also decreased on the 3rd week post-insult. This is juxtaposed by bull 4's embryonic score not being affected. It can be seen that cleavage rates on average decreased from 85% to 66% when piriforms, diadems, and vacuoles are over 50% in sperm samples. The researchers' answer to the question is not exceptionally strong. This study did show that scrotal instillation will have an effect on sperm morphology and embryo development, yet it also showed that not all bulls may be equally affected. The effect sizes were significant and are clinically important. For bull 1, normal spermatozoa decreased from 74.1% to 22.3%, and bull 3 normal spermatozoa decreased from 67.7% to 0.5%. As you can see in this table, the circle results represent a significant increase for each abnormality between pre-insult and post-insult. Bulls 1, 2, and 3 showed a significant increase at a p-value of 0.001 in amounts of piriform heads. But bull 2 had a significant increase in apical values at a p-value of 0.01. Bull 3 had a significant increase in diadems from 0% pre-insult to 50.2% 3 weeks post-insult. For bull 4, individual abnormalities does not exceed 10%. These studies show a strong relationship between abnormal spermatozoa and instillation, but one can argue that these results are biased towards bull 1 and 3, whereas bull 2 and 4 did not show such significant results. The main effect of bull had an insignificant effect on the cleavage rate, with a p-value greater than 0.05. But the sample collection date did have a significant effect, with the data being significant at a p-value of 0.01. From these results, it can be seen that there is a significant positive correlation between embryo development score and normal spermatozoa. The negative correlations between development score can be attributed to the escalating numbers of piriform heads, diadems, asymmetrical sperm, decapitated sperm, apical values, and severely abnormal spermatozoa due to instillation at different stages of spermiogenesis. It can be inferred the effect size of the expected data, increasing abnormalities with instillation, are significant, whereas the unexpected results, bull 2 and 4 having little or no effect, are due to chance, genetics, individual resistance, or scrotal instillation.
In conclusion, the scientific and clinical implications of this study is that scrotal injury will increase the number of abnormal sperm. Overall, this will decrease the rate of fertilization and embryo development. However, the fertilizing capacity of each individual sperm is still unknown. The abnormalities that thermal scrotal insult induce is piriform heads, apical vacuoles, and diadem. The extent of the damage may be correlated to the stage of spermatogenesis the individual sperms were at the time of insult. Usually, abnormal sperm are unable to penetrate the zona pellucida layer of the oocyte, resulting in low fertility. However, some seem to be able to do so. Additionally, sperm that appear morphologically sound may have abnormal chromatins, so can still penetrate into the oocyte but cause reduced embryological development. However, as embryological development is a multifaceted process, it remains unknown how each of these abnormalities affect all stages from fertilization to blastocyte formation, and therefore the fertilizing capacity of each individual sperm is still unknown. In relation to the veterinary profession, this information could be used to advise on breeds for particular environments and in bull evaluations, however not to compare between bulls. For example, a Black Angus bull in a hot climate may have detrimental effects on their semen quality and quantity, whereas a Red Angus in the same environment may produce viable sperm at a high sperm count. This method could also be used to determine the sensitivity of a particular bull to scrotal insult when using the control of pre-insult sperm from the same bull. However, this test cannot be used to compare two different bulls, as fertility is multifaceted and could be affected by genetic predisposition, diet, age, climate, and breed. Overall, this paper added to current knowledge. It was already known that abnormal head shapes, in particular piriform heads, would reduce fertility. This study was able to confirm this and provide additional information on the abundance of the different types of head morphological abnormalities caused by thermal insult and their effect on in vitro embryo development.